good morning, everybody. Good morning. Happy Monday. Thank you, Ronald. Let's get everybody back together after the weekend. Only announcement, tonight at 7 o'clock, new time. Your cyber sheet decisions are due, so make sure you get those in. Okay. Another announcement I want to make, although I think everybody's registered for classes and the clicker is having a bad day again, which is always fun, is that for folks who are interested in learning more about genetics and maybe thinking about grad school in this area, Dr. Shibano, who came and talked to you at the very beginning of the semester, he's our swine molecular geneticist. He teaches a course in the spring called Genome Analysis, where you look more into how the genome functions, how you use genetic tools that we have to identify alleles and variants causing specific traits, and then some comparative genomics, looking across species of how the genome functions. So if anybody's interested, I'd recommend that. It's Monday, Wednesday, I think 1 to 2:20 or something like that. Okay. All right, so last time actually in lecture, you're taking an exam, right? But before that, Dr. Lewis finished our discussion of multiple trait selection. Talked about several different methods that could be used. Tandem selection, you're selecting for a couple different things. You might want a threshold of each. Independent culling, individuals that don't meet a certain criteria down the road, right? And then he also gave you an example of a selection index. And if you remember, the index helps you weigh all those things, could be more than one, could be many, that are important to your program, right? Guys, you need your attention up front, please. Okay. So you'll be thinking about selection indices in lab. You'll do an example of that. Also in lab, if you push it hard enough, it works. We're going to work through examples of correlated response to selection. And here's this really pretty equation, right, on the board. I want you to think about, get your mind thinking about this again, and like I said, we'll do it in lab. Think about rate of change. In this case, we're thinking about rate of change of the trait Y. We're not selecting for trait Y. We're selecting for trait X. This is saying what's our rate of change of breeding value in Y due to selection for X over time. We're still selecting for X, right? So you have an intensity of selection for X. You have an accuracy of selection for X, just like that one you've done before, right? Only difference is we need this. What's that? It's our R. Come on, guys. What is our little R? We have a BVX, BVY, two different things. Correlation things. So R, correlation. R, correlation has lots of R's in it. That's how I remember it's an R. If we're interested in how Y is going to change relative to X, we need to know how they're correlated. If this is 0, selecting for X is going to have absolutely no impact on what happens for Y. But if they're highly correlated, selecting for X is going to have a higher impact on the change in Y, okay? Before, you remember this part, we had, when we were doing direct selection, we were interested in how much variation there was in that trait we were selecting for. In this case, we're interested in how much the other trait Y, changes. So we need to know how much variation is there in Y that is available for change to happen, okay? So this is putting together what you've been doing for a while, selecting for one trait, acknowledging that genes are pleiotropic. They have more than one effect, and many of our traits are correlated. So selecting for one thing, you're going to impact other things, okay? So that's really important in what we do as animal breeders. All right, so questions about that? Okay. Today we're going to start talking about inbreeding. We're going to talk about its impact on heterozygosity, and hopefully that you already have an idea of what that is. we'll put that in context to our genetic model. So thinking about inbreeding for polygenic traits. And then if we get to it, we'll start thinking about how we're going to calculate inbreeding based on our animals' pedigrees. Okay, so inbreeding, this is a reminder, I think we've defined it before, is the mating of relatives or mating of individuals that are more closely related to one another than the population is on average. Okay. So we should also be comfortable with the idea that inbreeding increases homozygosity. So in my example, I have a sire. There's his genotype at 2 loci. He gives that chromosome to his daughter. So she has an identical copy of it. She also has a big A and a little b prime allele. If I were to mate that daughter back to her sire, there is potential for him to donate that same chromosome with the big A and little b prime to that progeny. She could also donate that chromosome that originally came from her father to that progeny. And as a result, there's homozygosity in that progeny for the big A allele and for the little b prime allele. Okay? That's all we're talking about in inbreeding. We're talking about having some chromosome that starts from one parent getting passed down through a sire and a dam to a progeny and coming back together in the homozygous state. Okay, so no clickers. We're going to do the old-fashioned way with hands. So I want everybody to vote. I want everybody to vote tomorrow too as well, right? So this is practice. So inbreeding results in 1, establishment of uniform populations. Who thinks that might be true? I had an enthusiastic voter who changed their mind in the back. Not very many people? Okay, we'll talk about all of these. Does it cause improvement in traits of interest? Some votes for that. Okay. Neil's waving. Does it cause negative consequences? More hands for that so far than anything else. Okay. How about loss of diversity? And hands for that as well. Okay. So inbreeding actually, I'm going to argue, causes each of these things to happen. Oh, none of all the above. See, you're all just way ahead of me. Okay, so like I said, we're all going to argue together, since everybody just voted, thank you, that inbreeding causes all those things to happen. And that's something that you need to be conscious of. It's something that makes it exciting, right? Because we can get traits we want, but we also have to weigh the potential negative consequences. Okay, so some examples of each of those things. I think we'd all understand that our breeds, and it's kind of in their name, right? Inbreeding. Breeds are in a lot of ways inbred populations. So we start out with some individuals. We say these individuals share something we want, so we're going to keep breeding those individuals and their progeny. And in doing so, we get uniform populations. You ever heard of Herefords out there? They all look pretty dang similar. We have our Shorthorns, we have our Holsteins, which I think we could argue are almost clones of one another, that they're so inbred that look very similar. Same thing with dogs, right? Cats are always a little different, but cats are really good examples of this. Every single breed on the screen, and there's lots and lots more, were the result, the whole breed, it's a result of one individual that had a mutation. And cat breeders thought that mutation was cool, so they bred that individual back to his mother, to their siblings, to create a bigger population and create a breed. So inbreeding causes uniformity. Then you have a breed of all these cats that have curly hair, cats that have little folded over ears or really teeny legs, right? But it was a result of inbreeding. Similar with horses, right? So we get uniformity within our populations. We improve traits of interest. I'm a horse person, right? I'm going to give you horse examples for a lot of these. These are all quarter horses, all one breed. But within that breed, I think I've talked about it before, there's been selection for specific things. So horses that are good cutting horses that can work a cow, horses that are good racing horses, horses that stand and look really pretty and have big muscles, right? So within each of those types, even though they're all quarter horses, they've improved a particular trait that they want for that performance ability. This improving traits of interest shouldn't be a surprise, right? Milk industry is a really good example of it. Breeding practices have increased milk yield significantly, increased carcass traits that we want in our beef cattle, and then we've created breeds that have traits of interest, whether they have nothing to do with production or not, right? Okay, so those are two things. But negative consequences, Dr. Lewis talked about these examples in cattle, right? We have fawn calf syndrome, curly calf, developmental duplications. They're all due to a recessive, so it's homozygosity, which is in many ways our definition of inbreeding. Dogs, right? Breeders have something they want, which was the Merle mutation. It creates more white in the dog. If you want Merle, you're going to breed two Merles together. Turns out that if you have a dog that's homozygous for that mutation, they could have eye problems and hearing problems. And the example in horses we talked about, ovarolethal white syndrome, and this is HERDA, which is a skin problem in cutting horse lines. Those are both due to homozygous recessive mutations. Okay? So even though we're getting cutting horses that do really, really good job at what they do, there's some negative consequences, right? Okay. Finally, loss of diversity. And I've shown you this before in a little different context. In this case, let's say our 5 chromosomes represent individuals. There's a mutation right here that's yellow, and you like it, right? So pretend it's that little cat that showed up your door that had curly hair, and you decide, I want lots of kitties with curly hair. So you're going to keep breeding that cat, and hopefully those individuals are going to become more homozygous. And pretty soon, all of your cats running around the barn are going to have curly hair, which means, due to linkage to disequilibrium that we've talked about, not only is that mutation really, really common, so is a lot of the genome around it. Okay? So all those things happen with inbreeding. So inbreeding is very important. Finally, I want to remind you of this. We'll do an example. So previously we had this equation in our life, and it was a way to quantify, and we thought about this in terms of population, how inbreeding changes the levels of homozygosity. Okay? So we quantified the loss of heterozygosity as F. F's going to be a common letter we use for inbreeding. And we said that our F for a group of individuals is how much heterozygosity is lost from what we would expect over how much we would expect. Okay? So let's get you thinking about a little bit of Hardy-Weinberg and math. Let's say I have 3 genotypes. And I have frequencies 0.22, 0.36, 0.42. If our population with those genotype frequencies are randomly mating, and I asked you how much heterozygosity do you expect in that population, if I say that population's really, really big and randomly mating and there's no selection, there's no mutation, no migration, what would you tell me about what you would expect for heterozygosity? Hear whispers. Those hints I gave you, the big population, what does that click in your head? What were you talking about? Hardy-Weinberg, right? So if our population's in Hardy-Weinberg, how can I estimate the frequency that I should see of heterozygotes? It would stay the same from generation to generation, right? That's true. But how do I know if this is what I should expect to see if they're randomly mating? Remember that Hardy-Weinberg equation that we had? P's and Q's, right? So our frequency of our heterozygote, or we could say our big H genotype, right? For Hardy-Weinberg is 2pq. That is simply our expected heterozygosity, if everything's under Hardy-Weinberg. So to do that, we need to first find P and Q. So this is going back to a couple months ago, right? So if I need to find P, I'm going to say my P is the frequency of the big A allele. So that's going to equal 0.22 plus 1/2, 0.36, right? So 0.22 is our frequency of our homozygote. 0.36 is our frequency of our heterozygote, and they each only have one big A allele. So that's 0.4. Okay. My Q then, my frequency of my A, quickest way, right? 1 minus 0.4, 0.6. Okay. So then my expected heterozygosity, we just said if our good friends Hardy-Weinberg's law holds up, 2PQ, 2 times 0.4 times 0.6, 0.48. It's not what I have over there, is it? I have 0.36. So I want to know what my F is then. Expected. 2PQ, if Hardy-Weinberg was being upheld. My observed, which was 0.36. It's actually how many individuals in that population are heterozygous over the whole dang expected total. We get 0.25. So we should have 48% heterozygotes. We have 36%, which means that if we don't have as many heterozygotes as we should, they must be homozygous, right? So I was thinking about inbreeding like we did in the past with populations, with groups of individuals. So let's think about it now for individuals, right? So we're still going to call our inbreeding coefficient F. We're going to use a sub x for whatever individual x that we're interested in. And I'm defining that as the probability that two alleles at any one locus are identical by descent. Geneticists like to use IBD quite a bit, identity by descent. Let me get to my notes. Okay. So let's define that. So an allele is identical by descent if it has the same origin from both of those parents. So in this, our example from before, that sire had that big A allele. He gave it to his daughter, and this progeny is who we're interested in. That progeny's big A allele came from that sire originally, right? From that side. Progeny's other big A allele also came from that sire. It just was filtered through that daughter. So we call homozygosity like that identity by descent. That allele that individual's homozygous for came from some common ancestor back in the past. It could be a grandparent, it could be a great-grandparent, it could be even further back. But is filtered through both maternal and paternal sides to become homozygous in that individual. Okay? Does that make sense? Okay. If it doesn't, holler. Okay. So properties of our inbreeding coefficient. It's going to range from 0 to 1, not inbred, to completely inbred. And it's going to indicate the proportion of loci of an individual that are expected to have two alleles that are identical by descent. So similar to this F, when we were talking about the population, it's giving us a proportion of heterozygosity that's missing, or a proportion of homozygosity that we shouldn't see. Our F for our individuals is pretty much the same thing. It's saying how much identity by descent or homozygosity do you have, or do you expect at any particular locus of that individual. Okay. So that really depends upon how closely those parents are related. If that progeny has to receive a big A from both mom and dad, that only happens, like we said, if mom and dad are related. And it's more likely to happen if mom and dad are very closely related, right? So if an individual's parents are unrelated, our inbreeding coefficient of any progeny they have is going to be 0. So if they're unrelated, the sire and the dam don't share their alleles, therefore the progeny is not going to be homozygous. You can't get inbreeding. Okay, so we'll go through some calculations, but just kind of think about these numbers in your head. Inbreeding coefficients of progeny of various matings. So if you were to breed a parent to its offspring, an inbreeding coefficient of 25%. Think about how that kind of relates to that relationship that you think of between parents and offspring, right? So if you mate full sibs together, it's also 25%. If you mate half sibs, we half that. And I think this is pretty common in a lot of our production systems and our companion animals, right? We might have an individual, they share a single common grandparent, right? If that's the case, their inbreeding coefficient is about 3.1%. Let's put that in context of some of these things we talked about. So improving traits of interest, and back to my horse example. So I said that within those disciplines, even though quarter horses are a really, really big population, within those different disciplines, there definitely is inbreeding, okay? Ranges from pretty close to 0 in horses that are bred for reining to almost 4% in cutting horses. So we go back to that last slide, that means that cutting horses that were studied, they must share a grandparent on both sides of their pedigree, and probably a little more, right? Because if they share a grandparent, it should have been 3.1%. Okay. So our inbreeding helps get those horses to be what they wanted them to be. And who's heard line breeding? I think it's a fairly good common term, right? Some people say inbreeding is what is happening when something bad happens, and line breeding is when it turns out good. Okay? So line breeding is a method, an intentional method that breeders use to maintain something they like, right? I really like this particular bull. I'm going to keep breeding him. I'm going to keep breeding his daughters in hopefully a smart way to maintain what I like about that individual in my herd. Okay? So this involves mild inbreeding. And one of the really famous examples of this, especially in the horse industry, is what happened at the King Ranch. King Ranch in 1915, a stallion called Old Sorrel was foaled. Who's heard of Old Sorrel? My very first horse had him on her papers. So every horse, this is still true today, so that was 103 years ago. Every horse on their ranch today has Old Sorrel in its pedigree. So they set out to create a line breeding program. They said, we want this type of horse. He doesn't really look too fancy in that picture, right? He's pretty old. But he was a really, really good cow horse. And they said, we want to create a population of these horses to define our program and to define what we think a cow horse should be. So they actually did a really good job of keeping inbreeding to a minimum, but they still maintain Old Sorrel on the pedigrees of all their horses. So here's the current. Clicker's still not going to work. There we go. It's okay. Don't panic. So there's 3 stallions on here. The Boone, Marsala Red, and Keonos Moon. Those are the 3 stallions. They currently stand at their ranch. Here's Old Sorrel. Okay, so we could figure out how many times each of these stallions trace back to Old Sorrel. But they were very intentional in how they bred these horses to keep Old Sorrel in the pedigrees, right? So Wimpy familiar? Wimpy has the registration number of 1 in the Quarter Horse Association, the first registered quarter horse. Wimpy was a progeny of Panda, who was a daughter of Old Sorrel, and of Solus, who was a son of Old Sorrel. Okay? They really liked to mate Wimpy back to Old Sorrel daughters to create that line, and they did that quite a bit. By doing that, daughters of Wimpy, or foals of Wimpy, that resulted from mating of him with another daughter of Old Sorrel, were related to Old Sorrel at 62.5%. Think about how a parent is related to its offspring. What's that big R value? Right, parent, offspring. 1/2. Thanks. So, Wimpy's babies, when they were bred to another daughter of Old Sorrel, their relationship to Old Sorrel was 62%, bigger than 1/2. And that's the outcome of this line breeding program. Can keep more of that individual's genetics going than by outcrossing. Okay? That's kind of a fun pedigree. There's probably names you know in there. Peppy Sam Badger was one of their other very popular sires that keeps the Old Sorrel bloodlines going. Okay? Okay, so we said we inbreed to increase uniformity within breeds or lines. Many of our breeds are really inbred populations, so we shouldn't necessarily think of it in a negative context, right? Individuals share things. Our simply inherited traits are the easiest way to think of that. The polled locus in cattle, right? We want our whole population to be polled. It's safer for the cows and for the cowboys. That's a single locus. That's a simply inherited trait. By using inbreeding, line breeding in a smart way, we can make that happen. Same with coat color, those cats, crazy traits, right? Inbreeding isn't used as much in this manner, although I have an example just last year where I had a producer contact me who was doing this. But in the past, before we had really cool genetic tests, so you could test whether or not your animal carries a recessive, lethal, or bad allele, one way you could try to find out is you could mate that potential sire that you're interested in to a bunch of his daughters, right? So you're inbreeding on purpose to see if any of those progeny happen to have that bad trait or maybe that good trait that you're interested in, right? Because if that sire's carrying it and his daughters are carrying it, you should have some progeny that are homozygous, okay? So inbreeding can be done on purpose for that reason. And something we're going to talk about more in the coming few lectures is that by creating separate inbred populations, or even just separate breeds without thinking of them as being necessarily highly inbred, we can then use that homozygosity that differs between those two populations to enhance and increase our hybrid vigor or heterosis. Okay? So like I said, we'll come back to that in a little bit. Okay. So a little bit more about negative impacts of inbreeding. If we have homozygosity, we have the potential for those recessive alleles that might be bad to come together. Points I want to make sure I make is that inbreeding does not cause new alleles or does not cause recessive alleles to be bad. Inbreeding causes them to be manifest together in the homozygous state. Okay? So it increases the frequency and thus how often you see that particular phenotype. Okay, story time. Okay, so I'm going to take some time here to give you an example of this in dogs. And there's a reason why. One, if I have a chance to talk about cool genetics in animals, especially when they have videos, I'm going to do it because it's fun. But two, although we really want you to understand equations like that and calculating the probability of inbreeding in your animals, thinking about big A and big B and little b, this all has to do with the biology that underlies these traits. Whether it be a simple trait, where it's just one locus that's recessive and it causes something bad, or whether it's a polygenic trait, where there's recessive genotypes at many different places that influence what's happening. This all has to do with how it's actually changing actions of those genes in the genome to manifest that particular phenotype. Okay? So I'm going to talk about exercise-induced collapse, which is most common in Labrador retrievers. About 30% of Labrador retrievers carry a recessive allele that causes this condition. Just like quarter horses and Arabians and cattle, show cattle, Labrador retrievers have two really different types. That's a field trial dog. They're lighter bones. They look lean. They look athletic. That's a show dog. Some people call them the buoys when they are in the water, right? Because they're big and blocky. They're a little bit fatter. They're probably not as good at getting your duck out of the pond. Has anybody heard of exercise-induced collapse? Have you experienced it? Anyone? Okay. So exercise-induced collapse is what the name suggests. When these dogs are really, really excited, and everybody can probably think of an experience with the lab, you're playing fetch, right? And that dog will just play fetch all day long. They're going to wear you out, throwing whatever it is before that dog gets worn out. Okay? So an exercise-induced collapse, thank goodness for YouTube, right? This dog isn't even fetching. This dog is just running around. This is a black lab, obviously. You'll kind of notice those back legs. Maybe they're not working quite as well as they should. And we'll watch this dog run around for a while. Tail's still going. Dog's happy. Oh, start. Oh, oh. Right. So kind of dragging those back legs a little bit. Can we see that? Right? Still dragging himself along, right? The tail's still wagging, still happy to be out on a nice day, playing, but the back legs aren't working quite right, okay? So let's, one more example, another black lab. In this case, the dog's playing fetch. And again, I can make it bigger. Dog is happy as can be, but those back legs just aren't working. Okay. So probably not a good thing if you have like a hunting dog and you send him out into the water or something, right? And you want to make sure that it's going to be able to perform well. Okay. So our examples. So I won't go through the whole study, but they did what we've talked about before. The researchers did a linkage mapping study. They had dogs that were affected and dogs that weren't. And they identified this gene, DNM1, Dynamin1. And they found that unaffected dogs had at least one copy of the G allele. Affected dogs were all homozygous for a T allele. Here's that homozygosity that could be attributed to inbreeding. And so the question was, what does that gene do? And it turns out that that gene is expressed in the brain, the spinal cord, and it releases vesicles that move a neurotransmitter from one place to another to tell muscles what to do. So we're transmitting nerve impulses. And unfortunately, this video isn't working, and it's really cool because it looks like it was taken in 1960 something, even though it was in the 2000s. There's 2 vials of Drosophila. Okay, Drosophila are fruit flies. And they knocked out this gene. So they have fruit flies that don't have the gene and fruit flies that do. And then they just raise the temperature on them. So fruit flies that are normal, they're flying around their vial and they're just totally fine. Fruit flies that don't have the gene, the same one that causes this in Labrador retrievers, when the temperature gets to a certain point, they plunk, fall off the side of the vial. They can't fly anymore, they can't hang on to the side anymore. Their muscles aren't working. So very much like the Labrador retriever. So this homozygosity, this recessive genotype in fruit flies and Labrador retrievers, which is a really cool example of how genes do very similar things across species. What happens is that mutation changes amino acid. When the temperature goes up high enough in those dogs, and you could argue the temperature in those back legs are working pretty hard when they're running. The temperature raises in those muscles because they're working hard. That protein shape changes because of that mutation. Protein can't hold its shape like it should. And so those vesicles aren't transmitting nerve impulses like they should. Dog doesn't feel a thing, right? So you're kind of turning off those nerves, you want to say. It's not painful. The tail's not working as hard when it's running, so the tail still works, right? The protein's working in that tail. So all you have to do is give those dogs a break. Once they cool off, they'll be back to normal. Okay? So an example of how homozygosity changes the function of a gene, function of the protein that causes this condition. And I won't say that all labs that have this are inbred, but they share a common ancestor that had this mutation, right? Both their sire and their dam shared that common ancestor somewhere, so they were carrying that T, I think it was a T allele, that puppy inherited 2 copies of. Okay. So we're going to think about, in the upcoming weeks, more about our mating strategies, crossbreeding and outbreeding. And I want you to think about this case. So what if we wanted to breed Chesapeake Bay Retrievers who also have exercise-induced collapse? And a random sample of those dogs, Chesapeake Bay Retrievers. I mean, isn't that about the cutest puppy ever with little blue eyes, right? So the frequency of that allele in those dogs, so the N allele is our one that causes the collapse in the homozygous state. It's 0.13. The normal allele is 0.87. So hopefully we could all make a Punnett square. Oh, I have to have the right paper. Wrong paper. Say if I just mated Chesapeake Bay Retrievers at random, how many puppies would I expect to have exercise-induced collapse? We could set up a Punnett square, right? And we know that there's a normal allele floating around in the males and a bad allele, and we know that there's a normal allele floating around in the females and a bad allele, right? When we've done this in the past, we assume that big N and little n are at the same frequency, because we usually think about meeting one dog who can only have 2 alleles to another dog that can only have 2 alleles. In this case, we're thinking about a group of male dogs randomly chosen from the breed, a group of female dogs randomly chosen from the breed. So in that case, I have to consider these aren't at equal frequency, right? There's more of that big N allele, thankfully, than that little N allele. So I'm going to write out here to make sure I remember that those frequencies are 0.87 and 0.13. And then I can fill out my table just like we've done way too many times, right? Except that when I multiply my gametes, my alleles together from the male and the female, I also have to use their frequency to figure out what the expected frequency is going to be of each of those genotypes in the progeny. Okay. So in this case, I'm interested in how many are going to have little n, which is that recessive allele. It's 0.13 times 0.13, which rounds to 0.02. So 2%. Does everybody see that? Understand why we use these numbers here? These are the frequencies of those alleles in that population in general. Okay? We're going to be doing more things like this where we alter the frequencies in our Punnett square. So I want you to feel comfortable with that. Okay? Questions, let me know. Okay, so that was one case. So let's think about another scenario. So if I just made them at random, I only have 2% affected dogs. Maybe I can live with that, right? But let's say I have a male that I really, really, really like, and I'm going to mate him to random females, okay? So I'm going to stand my male dog as stud maybe, and then I'm going to mate him to random females. We have 2 alleles. Our females, what's the frequency of big N? .87, right? So I said they're just random, and that's the frequency of that allele in the population. Then the frequency of little n is 0.13. What about in this case now? What do I put here? 0.5, awesome. So if that's 0.5, that has to be 0.5, because that's a single dog. We're assuming in this case we have a male that we know carries it, So his alleles are at frequency 0.5 and 0.5, right? So then if I do the math, I have 0.5 times 0.13. I'm rounding again, but 7% of my progeny, of my puppies, are going to be affected. It's a frequency that we expect that allele to be in the females. The frequency, it's in the males. Final case scenario. I'm going to keep that same male that I like. Same male. We know he has two alleles. One of them's good, one of them's bad. And I'm going to mate him to his own daughters. His own daughters out of unaffected, homozygous unaffected dams. In that case, what would these frequencies be? I heard a point something. If this is dad, what percentage of his babies are going to get the little N allele? 50%, right? And we said mom is homozygous, unaffected. So mom's always going to give him a big N. So therefore, the frequency of this just depends on the frequency in dad, which was 0.5. Okay? So if we're mating that dad back to his daughters, we expect 25% of those puppies to be affected. So that is intentional inbreeding, line breeding, whatever you want to call it, right? And in that case, we have increased homozygosity for something that we really don't want. Okay? Okay, everybody happy? Relatively? Okay, so those are simply inherited traits. However, like we've said before, we're really interested in those polygenic traits. We're interested in growth, we're interested in fertility, we're interested in milk yield, we're interested in litter size. So our polygenic traits, they could have recessive genotypes due to inbreeding. So we have to think about how when we have recessive genotypes across the genome that affect those traits, how they in fact impact our phenotype, right? So together, all those different polygenic traits can influence our performance. Okay? We often think about that as inbreeding depression. So our inbreeding depression is when our inbreds have a decrease in performance. And most commonly, we see that in those polygenic traits such as fertility, survivability, and other growth and performance traits. Okay, so there's my inbred cat. Get it? Come on. It's like double inbred because it's already inbred because it's a cat and then it has bred. Never mind. Okay, so I want you to think about this. This should look familiar. We have our genetic model. In what part of this model does inbreeding depression come into play? Does it come into play in the breeding value, the GCV, the E, the environment, or maybe it doesn't play a role in the model? Think about it for a second. Chat with your neighbor. We're going to do a vote. Okay, come up with a vote, and we're going to have a vote here. Okay, ready? I want to see a lot of hands up when you think it's appropriate, and I'll ask you all four this time. So who thinks it comes into play in the breeding value? We have a few votes, good. Okay. How many thinks it comes into play in the gene combination value? More hands, okay. Environment. Okay, good. We're not talking about environment, we're talking about genetics, so that's a good thing. Or D, it doesn't play a role in the model at all. That's good, because that's kind of a trick question, right? Because if it didn't, we wouldn't be talking about it in genetics class. Move on. Okay. So let's think about our breeding value. Our breeding value is the effects of our two alleles, the additive effect, right? We have one, we get something. We have another one, we get something else. What's our gene combination value? What does it describe for us? Genes, are they not quite, but something about those two alleles. What about our two alleles at our locus? What does our gene combination value tell us about? Give me examples. What was that? Okay, so something with how those two alleles at the locus might interact, right? The relationship among each other. Is it a dominance? Is it codominance? So remember we said our breeding value is our additive effect of those alleles. Our gene combination value is how they interact, right? So inbreeding, we're talking about it with context of homozygosity and like recessive, lethal recessives. That's due to them interacting with one another, right? So in our case, our inbreeding depression is due to poor gene combination value. That's due to that homozygosity. Okay? So, 2 minutes. Let's think about this. Okay, so I'm going to give you a genotype of 2 different individuals. First, I'm going to define that my big alleles at each of 4 loci give me a value of plus whatever units you want to pretend they are. Okay? And our recessive alleles at each locus give us a value of -2 whatever. Okay? So if I have complete dominance, what is the value of my big A homozygous genotype if each allele is worth 4, 8, right? If I have complete dominance, we're just thinking about one locus, but each of them have the same values, right? If I have complete dominance, how does the value of my heterozygote compare to that of the homozygote? Same, right? It's also +8. And if I have a recessive genotype, then I have 2 little a alleles. They're each worth -2. And -2 is -4. Okay. So say I have an individual that's heterozygous for each of these alleles, and I have an individual that's homozygous big A, homozygous little b, homozygous big C, and homozygous little d. Okay? If I think about my breeding value, I'm just thinking about how much each of those individual alleles is worth, right? So I say, how many dominant alleles do I have? Because they're each worth 4. So I'm going to multiply 4 by 4, right? 4 times 4. And then I also have 4 little recessive alleles, and those are each worth -2. So my breeding value turns out to be +8. Is the breeding value of this individual any different? How many dominant alleles do I have that are worth +4? 4, right? 1, 2, 3, 4. They're not the same, but their value is the same. So So in that case, I have two totally different genotypes. I have a lot of heterozygosity here. My breeding value is 8. I have a lot of homozygosity here. My breeding value is still 8. Okay? So ponder how that homozygosity is going to change our gene combination value, and we'll pick up on that on Wednesday. The dog's still playing.